The goal of GenomeDiver is to help improve our success in making diagnoses from genomic testing. The current problem is that clinicians have very limited means of communicating their insights to colleagues in genomic diagnostic laboratories. GenomeDiver is designed to facilitate this communication. We start in the genomics diagnostic lab. Once they have completed sequencing, they have a list of DNA sequence variants to go along with the information describing the patient's phenotype received with the test requisition. Typically, the phenotypic information is captured as human phenotype ontology, or HPO terms. They initiate a dive by loading their sequencing and HPO term information into GenomeDiver. GenomeDiver starts by running an analysis to identify the DNA sequence variants most likely to be associated with the patient's HPO terms. Other HPO terms that can be caused by these variants are now of interest in helping to make the case for one of the variants causing the patient's problems. The clinician is then prompted to join the dive. They are shown a list of HPO terms and asked to categorize them as present or absent in the patient, or otherwise uncertain. Using genomic information to prompt the clinician to look for phenotypic features is what we call reverse phenotyping. This typically takes 3 to 4 minutes at most per patient. GenomeDiver can now use this enhanced list of HPO terms to reanalyze the genomic sequence information, ranking variants and genes and associated diseases. The clinician is now presented with the top-ranked genes and diseases with links to explore each to see whether any look like a good fit for their patient. Diseases of interest are flagged, and the clinician is given the option to write a short note to colleagues in the diagnostic laboratory. Typically, this takes 2 to 3 minutes at most. GenomeDiver bundles the refined list of HPO terms, any disease flags, and comments from the clinician and returns the information to the genomic diagnostic lab. The diagnostic lab colleagues now have an enhanced description of the patient's phenotype and the clinician's insights to help them to understand the patient's genomic sequence variants. Our goal is to engage the clinician for no more than several minutes so that we can increase our diagnostic yield from genomic information, enhancing clinical care.